Today we're kicking off our introductory microbiology course, and we'll be going through the Tortora microbiology textbook from Pearson Benjamin Cummings. Our emphasis will be on human health and disease as opposed to the environmental microbiology elements that are found within the book as well. As we'll be discovering, there's an almost unimaginable number of bacteria living in our gut, in our GI tract, especially in the colon, large intestine, and they provide some very important services to the body. And there's just a picture, false color scanning electron micrograph of some of those bacteria. And we'll be talking more about microscopy specifically in another chapter. So what are microorganisms? They're any living thing that's too small to be seen with the unaided eye, and I shouldn't have said living probably because we're going to look at viruses as well, which technically aren't really alive, but they definitely cause human disease, so we'll be taking a look at those. Here's some pictures that represent different types of microorganisms. Here we have bacteria in the upper left, and we have some fungi, some mold over here in the top right. We have a protozoan, a single-cell animal, over here in the bottom left. An alga, or algae is a plural, is a type of organism that we'll be taking a look at, a classification. And finally, over here on the bottom right, viruses. So microorganisms are necessary for life on Earth. They're responsible for decomposing all of the plants and animals after they die. And if it weren't for microorganisms, the planet would be littered with such a deep pile of organic material, there'd be no way for things to persist, living things to persist. So they break down everything, and they recycle all the nutrients that become available for additional organisms to exist on the Earth. They do all kinds of good stuff by way of industrial manufacturing products. They are involved in producing all kinds of foods that we take for granted. So microorganisms are super important, and only rarely do they actually cause disease. Very few microorganisms among all of the panoply that exists are actually disease causing. For the most part, they're good and useful and productive. So, for example, what do we gain by learning about microbiology? And again, mainly we're going to be talking about our ways of protecting ourselves from disease, from infectious diseases, and we've come a long, long way. As we'll see, we'll do a little history investigation in this chapter, and we'll see how far we've come in terms of our understanding of microorganisms, but also there's all kinds of things, again, that have happened by way of what we eat and what we manufacture that have been perfected by our understanding of microorganisms. Biologists love to classify things, group things, and make it easier to organize our thoughts about them and what we learn about them. And when it comes to microorganisms, Carolus Linnaeus developed a naming system in which each living organism has two separate names, a genus and then a specific epithet. So as we'll see in a couple of examples, you'll be very familiar what I'm talking about there. When we express these things, we always italicize the name of a microorganism, a bacterium, for example, and they have these kind of Latinized sounding names, and they're agreed upon throughout the world so that we can communicate efficiently about the microorganisms we're talking about. So these names are not just random and arbitrary, but they are agreed upon by the scientific community throughout the world. Here's an example, Staphylococcus aureus. And Staphylococcus is the genus name of bacteria that which there are a number of different species or subtypes. And aureus stands for the appearance of the colonies that are produced as these bacteria grow. They have a gold color colony, so aurea is a prefix that means gold colored. Escherichia coli, very, very familiar bacterium. The genus name comes from the name of a scientist who discovered these organisms, and then the epithet refers to where they live, the colon. So we see that oftentimes the name honors a scientist of some historical importance. All right. A lot of times we will abbreviate. You'll see in this course we'll many times abbreviate the names of bacteria and fungi and so forth by using the first letter of the genus capitalized and then the epithet. The important thing to talk about at the beginning in this regard is don't start doing that too soon. It's better, I think, probably for the first few chapters even to always write out the entire name, because there are so many different genre, I should say, that have the same first letter that it can be very confusing until you really get to know them by heart. So I recommend sticking with the whole name, but again, eventually you'll see Staphylococcus aureus will be just denoted S. aureus because it's quicker, shorter. All right, let's take a look now just briefly at those different types of microorganisms that I mentioned out of that first slide of pictures, and maybe identify some characteristic features of each one of those. Bacteria are what are called prokaryotes. They're organisms that don't have a nucleus. They have a cell wall, however, and it's constructed predominantly of a material called peptidoglycan. That just means protein with polysaccharide together in kind of a mesh. So peptidoglycan is what bacterial cell walls are made out of. They vary quite a bit in their construction, as we'll see. But that's the general stuff. They divide by binary fission, and they produce two identical offspring. And when these divide, we'll take a look at that in a later chapter also. They have a very broad range of metabolic capabilities. You've probably studied mainly animal cells in your experiences in biology so far, and also some plant cells. And in general, the metabolism of all those cells is similar. They use, for example, most animal cells use glucose for a source of energy and a source of organic material to build out of. And it turns out that bacteria are extremely diverse in being able to use a number of different sources for both their carbon as well as for producing energy. Some bacteria, for example, are photosynthetic. They can capture CO2 from the atmosphere and fix that to make polysaccharides. They can also sometimes use it just to produce energy, use photosynthesis to produce their energy that they need. So we'll see a lot of different examples as we go through later on. Here's just a micrograph, a scanning electron micrograph of Haemophilus influenza. So there's the genus and species there. It's just one type of bacteria that can cause pneumonia, infections in the lung. It's interesting, I think, because this microorganism was named for its discovery during a time when the scientific community was intensely interested in solving the problem of what caused the 1918 influenza pandemic. And there was a period of time when they thought this bacteria might be responsible for that disease, but later it was found that it wasn't, as we now know, the influenza virus, but hence the name influenza. Archaea are also prokaryotes, and in that way they're a lot like bacteria, but instead of peptidoglycan, they have a different type of material that makes up their cell wall. They do have a cell wall. Interestingly, Archaea are known for some of which are able to live in very extreme conditions, extremely salty environments like Great Salt Lake, extremely hot temperatures like hot springs. Old Faithful comes to mind, and out west, and a very popular mecca for summer travelers. So these things can live in very extreme environments, but there's not too many, very few human diseases caused by Archaea. We won't mention them probably anymore during this treatment of our introduction to microbiology. I will mention briefly, though, that methanogens are species that produce methane gas as a byproduct of their metabolism, and there are methanogens in your intestines that produce methane gas, as most people are fairly familiar with. Flatulence comes in part from methane. A lot of it comes from methane produced by Archaea living in your gut. Fungi Now we switch from prokaryotes to eukaryotes. Eukaryotes have a membrane enclosed nucleus where their genome is located, all the chromosomes, and they also have membrane enclosed organelles in the cytoplasm of the cells, so they're a little bit more complex in some ways. Fungi have a cell wall also, but it's made of chitin, and chitin is a different version of how to build a cell wall. It's made of glucose molecules that have been modified chemically, and then it's assembled into polymers. It's a very specific kind of material. I had written out a shock and awe value. I don't know if I can find it now. Um, there it is. It's in the cell wall of fungi and also of certain insects, and something like 1 billion tons of chitin is produced every year by all these microorganisms. Isn't that interesting? So you may not have heard of chitin, but now you know it's extraordinarily prevalent. Um, more about fungi. Molds and mushrooms are multicellular versions, and they consist of thread-like assemblages of cells called mycelia, and they are then woven or wrapped into filaments called hyphae. And they almost resemble the roots of a plant when they sit in the ground and they extend their hyphae down into the ground. Yeasts are unicellular fungi, and a lot of fungi can exist in both unicellular and multicellular form under different conditions. So we'll be taking a closer look at that in a later chapter. Protozoa are single-celled organisms, single-celled animals, and they ingest organic materials that they use for their energy and for building material. They may have motility. There's three sources of motility for protozoa. They may have pseudopods, in which you can see this amoeba can produce extensions, arms that can reach out around a particle that it wants to ingest and bring it right into the cell. Those are called pseudopods. Cilia are motorized little hairs on the surface of an organism, and some protozoa can move very, very quickly in water, as we'll see with cilia. Also flagella. We'll learn more about flagella, just another type of motility organelle or instrument on the cells. And here we can see an amoeba about to engulf with its pseudopods, these arms, around a food particle it wants to ingest. Algae, here's an assemblage of cells that make up an alga, particular type, and they're often photosynthetic. And they may range anywhere from seaweed down to single cell algae that have various interesting complex life cycles. They produce a lot of useful compounds that we use in the microbiology lab, as a matter of fact. Viruses are the smallest. They are not cellular. They don't have any organelles. They have basically a genome. All their genes in the form of nucleic acid, either DNA or RNA, are wrapped up in a protein coat, and then that may or may not be surrounded by a lipid bilayer, an envelope, it's so-called, that came from the cell from which it emerged. Viruses can only replicate inside of a living host cell, so they find their way in and they take over the machinery of a cell to produce more virus particles, and then they escape either by blebbing off or by bursting out of a cell. Multicellular animal parasites. Helminths are a type of parasitic worms, and we'll be talking about some very, very awe-inspiring helminths. We don't have too many parasites where we live here in the United States, at least not too awful many, and so we don't really learn too much in grade school about them or anything, and we'll be talking about some very intriguing organisms that most of the world is all too familiar with, that invade their everyday lives. The worms can be quite large, very, very large indeed, in fact, but the reason they kind of probably find their way into this discussion of microorganisms is because they have at least some part of their life cycle is microscopic. So we're going to sneak them in here, talk about them in microbiology. Here's a little a picture of a parasitic guinea worm being removed from underneath the skin of this particular person. As I said, these are very, very common experiences in much of the world, and the way to remove that particular worm is to pull it out by wrapping it around a stick, and it's thought that this symbol for the medical profession, this rod of Asclepius, might represent or be derived from this problem of having to get these guinea worms out of the skin of its victims. So more about classification. Carolus Linnaeus, we saw, gave us a naming scheme to identify specific microorganisms. All microorganisms, all living things for that matter, can be classified into 3 domains. Carl Woese, W-O-E-S-E, is the person credited with developing this 3-domain system of classification of microorganisms, as well as all plants and animals. And many schemes for organizing or classifying plants and animals and microbes have existed over the years and have gained traction for periods of time, but this is currently the most popular, probably, classification system, so it's the one we'll be talking about in this class. It's pretty much been accepted by scientists of our day. And here's just a little way of demonstrating the relationship, the relatedness of various members of the bacteria, the domain bacteria, the domain Archaea, and the domain Eukarya. And you can see that they're thought to have an original ancestor that over time has changed, if you will, if this is the theory that many scientists accept. And as they changed, they branched off and acquired different traits and lost other traits. For example, you can see right here where this line is pointing, mitochondria are organelles in eukaryotes that many scientists believe came from bacteria and chloroplasts that entered into larger cells and eventually took up residence and became part of the normal organelle panoply within the cell. So mitochondria are thought to have derived originally from bacteria that were free-living and wound up inside of larger cells. Interesting, isn't it? And for example, in the same way, chloroplasts winding up inside of plant cells, giving them the capability of doing photosynthesis. Anyway, here's this branch. Giardia is a microorganism that branched off here and lost its mitochondria. So many of these organisms here may not have any mitochondria. Many organisms do. Now let's take a quick look at some historically important periods relating to microbiology and history, and also some scientists who have been credited with some major turns of events within the history of microbiology. Very cool stuff. Robert Hooke was the first one to describe cells, looked in a magnification of some cork and saw little boxes which he identified as cells. And that was the beginning of our understanding the cellular basis of living things. Antony van Leeuwenhoek in the 1600s discovered a way to grind small lenses. You could take pieces of glass and grind them into a particular sort of an ovoid shape, or I don't know what the right word is, that would allow magnification of light and focusing of light so that small objects could be observed in a larger appearance. And he would encase them in these brass structures, these little microscopes, handheld microscopes that he created. And this is a little facsimile, a replica of a Van Leeuwenhoek microscope with a tiny glass lens. And he would put a sample, a drop of something or a chunk of something that he would gather either famously from scrapings of the teeth or from water droplets or from decaying broths produced by taking plant matter and sticking it in water for a period of time. And he observed for the first time microorganisms. And he's a very intriguing guy. He was actually a merchant who was interested in particularly fabrics of various types, and he needed a way to magnify fabrics to take a look at the quality of the fibers that made up the fabrics, and he then stumbled upon this observation of microorganisms, and he became fascinated with it. And he made literally hundreds of these microscopes, which he would use only once. He would put a sample on there, and he would then set it aside to look at later. and then create another one. And he just kept building more and more of these microscopes. He didn't share his discoveries with anyone. He made very elaborate drawings, books of drawings actually, of all his discoveries, and he presented them to the Royal Academy of Scientists in London, and introduced the scientific world to microorganisms, this merchant, and yet he didn't share any of his technology with anyone. No one looked in his microscopes but him, and when he left this world, he left all of his microscopes went with him, and none were retained. So anything you see that by way of a Van Leeuwenhoek microscope, it's a replica because none survived. And there was a very long period of time before any good microscopes were produced since his death that would allow people to observe microorganisms once again. Virchow is credited with the cell theory, the idea that cells arise from pre-existing cells, and that seems obvious. Yes, of course they do. When cells divide, more cells are produced. That's second nature to us. But the thing is, at the time that he lived, there was a battle raging over the idea that life could arise spontaneously from nothing, the so-called vital force. And many people took it for granted because, think of it, they would take fresh food and put it out in the room, and over a period of time, it would begin to decay, and it would go bad, and eventually, and some microorganisms may be found in there, maybe even some bugs, and it just looked like it appeared out of nowhere. And this idea of spontaneous generation was a well established notion. Biogenesis is the competing idea that all living organisms arise only from pre-existing living organisms, and they don't spontaneously appear out of nowhere. And there was a great big debate that went on for many years over this subject. And I just want to run you through some very fascinating observations that were made by various scientists along the way, trying to establish which is true, spontaneous generation or biogenesis. This fellow Reddy discovered or demonstrated that if you prevented access by flies to meat that was put out and allowed to decay, no maggots would appear. So the maggots weren't just arising spontaneously out of that so-called vital force. They had to come from flies that landed on the food first off. So that was in support of biogenesis as the source of the maggots. Needham took some broth, and they knew that if you heated broth up, you could kill all the microorganisms. So he took some broth, which just, again, this is a rich water-based soup made from organic material that had been placed in it and allowed to kind of go a little bit, decompose a little bit, and then he would heat it and kill the microorganisms. And then he took his heated broth, which we know has no living microorganisms in it, and stuck it in a sealed container, and there was microbial growth. And this suggested, oops, we've made a mistake. Really, there is spontaneous generation. It's been demonstrated here, and for a while this kind of put the brakes on the modern understanding of biogenesis as the source of all living things. But it was understood later that he contaminated his samples when he transferred the heated broth from a pot on the stove into some flasks to seal them up in. He didn't understand that the flask already had some microorganisms in them. So this guy, Spallanzani, instead heated his broth within the flask in which they're going to be contained, and sealed them, had a little glass neck that could be melted closed and showed there's absolutely no microbial growth. And so that was kind of in favor of biogenesis then. Nothing grew in the already sterilized broth. There's no spontaneous generation of any microbes. But then some people started to complain that, well, for some reason it doesn't work. The vital forces aren't able to function without having the container be open in some way. And so there was more experiments done, more debates. But Louis Pasteur is the one that eventually, I'll skip right over the slide to the next one, and he once and for all demonstrated that there's no such thing as spontaneous generation. He placed a rich broth into a glass flask you see in this picture, and then affixed onto it a thin glass tube or neck, and shaped like the letter S right here. And then he boiled the the broth within the flask and killed all the microorganisms, and then let the flask sit indefinitely, however long, even years of time, nothing would grow in that broth. Whoops, I lost myself. That's okay. You don't need to see me, I don't suppose. The point is that even though it's open to the air, there's no microorganisms appearing by spontaneous generation in this boiled broth. Interestingly, previously, if he opened up and left the glass tube sticking straight up, then dust falling from the air down into the broth would contaminate it, allow it to grow, and it became known that there were microorganisms floating around, but not spontaneously arising within of sterilized broth. So that was a pretty cool demonstration of Louis Pasteur, somebody who contributed a whole bunch to our understanding of many, many things, including microbiology. For example, he discovered the idea of fermentation. In the time that he grew up, wine was a very, very important drink of choice for many, many Europeans where he lived. And to manufacture wine, sometimes things went well. You got a nice wine with a little bit of ethanol in it, and sometimes you didn't. You got a really sour tasting material with a lot of acetic acid and vinegar. And he discovered that it was microorganisms taking the alcohol and converting it by what's called fermentation over to acetic acid that would ruin the wine. So microbes were responsible for it. And so that was a big breakthrough in understanding how to ferment wine without getting the bacteria in there that would ruin it. Along the way, among his many contributions to industry and microbiology is the idea of pasteurization. In many manufacturing processes, pasteurization is used, a method of heating something up to a sufficient temperature to kill most microorganisms just long enough for that process to take place and not ruining the particular product, such as pasteurization of half and half cream that you might put in your coffee. It's been subjected to, it's milk basically that's been concentrated and subjected to a high enough heat that for a period of time you're going to have it in your refrigerator, it's not going to go bad. If you put a gallon of milk in your fridge, you've got it so many days before it's going to grow a culture of bacteria and not taste very good anymore, while your half and half can stay for a long, long time in the fridge because of pasteurization, a way of killing most of the microorganisms that normally, microorganisms are what spoil our food and drink oftentimes, and so we're going to kill a lot of those bugs and prevent that spoilage from taking place. So we're making good progress in understanding microbiology and applying it to various good uses in the world by this time. And we come to the golden age of microbiology. Things really took off with the understanding of microbes and what they're all about. Remember, this is all fresh stuff and all fresh discoveries. People knew nothing about infectious disease or microorganisms as the basis of disease. Semmelweis was a guy who was working in a hospital in which physicians were going from patient to patient to patient to deliver babies and cross-contaminating their patients. There was a tremendous incidence of death of women in the period of time, the puerperal time, that's a period of time, the few months after delivery of a baby, and the people were getting infections and dying. Well, they didn't know they were getting infections. They were getting sick and dying, and it was a terrible, terrible thing. And so he instituted this idea of hand washing in between the processes of delivering babies, or between medical students or doctors working, teaching labs with human cadavers, and then going right into deliver babies with the same hands with stuff all over them. He instituted hand washing, which hugely diminished the incidence of puerperal fever, the death of these women from infections after their pregnancies were over. Joseph Lister was a guy who was a surgeon, and again, surgery was a very, very iffy thing. It was a last resort kind of thing to treat disease or any problems with humans because so many people died of infections after surgeries. Joseph Lister found out that if you used a strong alcohol, an acidic alcohol called phenol, and slathered it around the wound opening when he did the closing, then his patients didn't die of infections. And so it was a disinfection, means to remove microorganisms from something using a chemical agent of some sort. So that was a big, big breakthrough in surgeries. Robert Koch, we'll talk a lot about him because huge contribution specifically to microbiology. He was the one that demonstrated a specific microorganism could cause a specific disease. Sorry about all these pop-ups happening. Um, so Koch's postulates are a series of steps that are used in any given instance if there's a new disease discovered and it's thought to have a microbial basis. If you follow Koch's postulates, a series of steps, you can demonstrate a specific microorganism that might cause that disease. So that's what Koch's postulates are, a series of steps used to, word oftentimes used proved. That isn't technically scientifically good to say, but demonstrate that there's a unique microorganism causes a specific disease. So that's what Koch's postulates are. We'll talk more about that. We'll list those steps at another time. Vaccination. I should probably speed up this whole discussion a little bit. Vaccination. Edward Jenner is credited with inventing the idea of vaccination. He infected someone intentionally with cowpox and demonstrated that they were then immune. Immune means not susceptible to infection with smallpox. Smallpox was a horrible virus that was throughout the world that had a tremendously high mortality rate and just a very, very terrible thing. And the development of this vaccine changed everything, as did the whole idea of vaccines changed our lives forever. And that infection, that virus has been essentially eliminated from the world since. But it's but that discovery of the smallpox vaccine was hugely important. Chemotherapy is the idea of treating diseases with chemicals. Let's say we have a disease and we know it's caused by a certain microbe. If we can find a chemical that will kill that microorganism but not kill the host, well, then we've got something, and that's called chemotherapy. We use the word chemotherapy usually today to mean treating cancers with chemicals, but in truth, chemotherapy just means treating any disease with chemicals. And antibiotics specifically are chemotherapies or chemicals that will kill bacteria or fungi, and they come from bacteria or fungi. So they're naturally occurring chemicals that have been discovered, already produced by certain microorganisms that can then be purified and used to cure infectious disease. Another example, quinine from a tree bark used to kill malaria, get rid of malaria parasites in someone's body. Paul Ehrlich is credited with using the term magic bullets. He was looking for a treatment for syphilis, a venereal disease that's a bacterial disease, and he developed an arsenic drug that would kill the syphilis bacteria and cure that disease. And he coined this term magic bullets, finding a specific chemical agent that will kill or at least fairly specific, kill a microorganism to cure disease. Sulfonamides or sulfa drugs were invented in the 1930s, but they didn't really come into prominence as a treatment for disease in the way that penicillin did. Alexander Fleming discovered penicillin by accident. He was working in a microbiology laboratory and growing bacteria. You can see bacteria growing on an agar plate. That's the substance we isolate from seaweed to grow our microorganisms many times on, as you'll see in the microbiology lab. And it got contaminated with a fungi, a mold that got onto the plate. And he noticed that there was a zone around the colony of fungus in which the bacteria could not grow. And he was genius is knowing when you've discovered something, and that's what we have here. He realized there's something of that penicillium fungus that may be able to help people, and in fact, penicillin was isolated, and then it was the first actually mass-produced and readily available antibiotic for treating disease. We'll talk a lot more about that. Bacteriology is just a term used to describe, obviously, the study of bacteria. Mycology would be less familiar, the study of fungi. Parasitology, when you're talking about those multicellular organisms that we'll be talking about later, the helminths and things. Parasitology. Genomics is the study of the genes of an organism, all of its nucleic acids that have the recipes for producing proteins, those are its genes. Genomics is the study of genes of an organism. Immunology is the study of the immune system and its ability to protect us against infections. And we'll have a couple of chapters to talk all about the immune system of the human body. Virology, studying viruses. Genetic engineering. Genetic engineering is what's the term that's used to describe manipulating genes in order to produce organisms that will be helpful to humans. So in the 1960s, Paul Berg, for the first time, took some DNA from an animal cell and inserted it into a bacterial genome, the genes of the nucleic acid, the DNA of a bacterium, and showed that the bacteria would then produce an animal protein. So this idea that DNA is the recipes or the information for producing protein in cells. So that was a huge breakthrough. Molecular biology is the studying that relationship between DNA, RNA, and proteins and the way they're produced in cells. Biotechnology is using microbes for producing products that we find useful. As we said earlier, there's a whole bunch of foods that are produced by fermentation, cheeses and yogurts and so forth. But there's all kinds of industrial chemicals like acetone that are produced by biotechnology using microbes. Genetic engineering has opened up biotechnology in a big way because we can genetically modify microorganisms to produce products that we're interested in that they didn't produce before. So now we can either produce insecticides, for example, in microorganisms, vaccines. We can produce vaccines by inserting some genetic information in a microorganism that we want to use for making a vaccine. We can add enzymes to make a microorganism have different properties or actually deliver a gene. We can deliver genes into other cell types. Deliver missing or defective genes can be cure disease. That's called gene therapy. Or we can modify some plants so that they have new properties like insecticide properties or produce vitamins that they didn't used to produce before. So all kinds of cool stuff going on in biotechnology on the basis of genetic engineering. Already shown in the previous slide, recombinant DNA technology means taking genetic information, nucleic acids from multiple sources and connecting them together to produce something that's useful for DNA, for biotechnology. Microbial genetics, studying how microorganisms inherit their genetic information. And as we're going to see later on, it's a pretty important thing because we've discovered that microorganisms can share genetic information horizontally, and genes from one bacterium can be acquired by acquired by other bacteria right in the same generation, which makes spreading of disease-causing agents pretty scary. Genomics, we already mentioned that. Recombinant DNA, we've already defined that. I need to tighten up that slide there, I guess. Microbiota, you hear this term a lot, probably even in the news. Microbiota refers to the microorganisms found in and on a person's body. And in your gut, for example, as I said, there are just unimaginable numbers of bacteria growing in there. And those bacteria are very useful because, for one thing, they are so voluminous and numerous that they prevent disease-causing microorganisms that we may eat in our diet from ever being able to grow to sufficient quantities to produce disease in our bodies before they're exited the body by excretion of feces. And so these normal microbiota are very, very helpful to us in that way. They also metabolize a lot of nutrients that we can't metabolize, like cellulose from plants can be broken down and turned into sugars that our body can use in the gut. We don't have any enzymes to break down cellulose from plants, and in addition, making vitamins. So vitamin B12 and K come from microbes in your colon, and then those could be absorbed and made good use of by your body's cells. So a lot of good things happening with normal microbiota. We have a lot of, we'll talk in the immune system, a lot of ways that your body can resist or be resistant to infectious disease, including your acidity of your skin surface and your stomach acid that kills microorganisms. We'll spend a lot more time talking about that later on and so forth. Here's an important topic, just quickly introduce biofilms. Um, microorganisms, bacteria, when they grow to a certain um, density in a particular environment where there's a lot of nutrients for them, they communicate with each other and they begin to secrete some polysaccharides, some sticky sugar substances that will then enclose them, and you'll have a whole community of bacteria living within a mass of what's called a biofilm. And biofilms can grow on organs in your body. They can grow on medical implants, catheters. They can grow on pipes in the plumbing system. They can grow on rocks in a stream. But biofilms provide a haven for microorganisms to live and make them very inaccessible to the immune system or to chemotherapies. So we're going to find out that biofilms are a real big problem in immunology and medicine. Emerging infectious diseases. When a microorganism overcomes the resistance that your body has, such as the inhospitable nature of your skin, or the acid in your stomach, and all kinds of properties of your body to prevent disease, sometimes microorganisms can circumvent those things and overcome the resistance and cause disease. Well, there's a lot of newish microorganism-based diseases, infectious diseases, arising in the world, and I will mention just a few of those. Various encephalitis viruses are becoming more and more prevalent, and they're starting to cause some scary diseases. Encephalitis means the virus can infect the surface of your brain. And so West Nile virus is spreading throughout New England and different parts of the country. It's carried by birds, and it causes encephalitis, and it can cause severe disease and even death very occasionally. Here's a mosquito is the vector that transmits the West Nile virus as it takes a blood meal. It also transmits the virus from one host to another. Bovine spongiform encephalopathy. A prion, or prion, depending on your choice, is just a protein, a chain of amino acids that can infect a body and cause disease. Can you imagine? That's all it is a protein that somehow can be ingested. So if a cow has bovine spongiform encephalopathy, and someone consumes some part of the nervous system or material from the nervous system that's been left over from butchering, consumes a prion that was causing that disease, then that prion, that protein, can find its way into the nervous system of the human and start causing degeneration of the brain and central nervous system. And that's called Creutzfeldt-Jakob disease. And that's just a horrendous thing. But just the idea that a protein cell alone can be an infectious particle, and it's becoming a problem more and more. There's a picture of some nervous tissue that's been partly degraded. The neurons are being killed by this prion. We'll talk more about what's happening there. E. coli. Now, one thing I want to dispel is this idea that when someone quote unquote gets E. coli, now they're sick. E. coli are present in your colon in vast quantities, and again, they're part of your normal microbiota. They provide a service to your body, multiple services to your body, so they're good. But this O57H7 refers to specific E. coli strains that have acquired genes that allow them to cause disease. They produce a toxin which is harmful to the lining of your intestine, and so E. coli that cause disease have become an increasing problem. You may hear every summer of some people that got quote unquote got E. coli or food poisoning from E. coli from hamburgers at a fair or something like that. Well, it's usually E. coli O57H7, and we'll talk more about what that exactly means to cause disease. And there's some E. coli that are going to cause hemorrhagic disease. They're going to kill the lining cells in your epithelium of your intestine and cause bleeding to happen right there on the surface. Pretty terrible. Group A, Streptococcus. Streptococcus is just a genus of bacteria, but there are numerous different species or types of Streptococcus, and some of them cause some very prevalent diseases, and some of them are becoming more and more prevalent all the time. They're an emerging infectious disease causing havoc in our medical community. You probably heard of Ebola. just a year or two ago, Ebola caused a fairly big outbreak in Africa. There had been an Ebola outbreak years ago. There was actually a movie produced, a book written and a movie produced called, um, uh-oh, I forgot the title of it now. But anyway, there was a very famous situation. Ebola, hemorrhagic fever, is a disease with a very, very, very high mortality rate. So high that it was thought that Ebola would never produce a dangerous kind of epidemic because it would kill its host so fast that it would never have a chance to spread. Well, that's been ruled out because some very bad epidemics have happened in Africa over the last few years, in which many, many, many people were killed, including healthcare workers, because again, this very infectious disease that has a very high mortality rate. Influenza, avian influenza, is becoming more of a problem. Just this year, hundreds of millions of domesticated animals, waterfowl, poultry, had to be sacrificed because of an outbreak of avian influenza. So the problem with influenza types is that there seems to be the ability of different strains of influenza to share or swap genes such that brand new influenzas that have different host ranges, meaning they can infect different organisms and have different degrees of virulence or disease-causing capability, emerges spontaneously. So the idea that these influenza viruses that are very similar to human infecting influenza viruses is not cool, and so we tend to take very seriously on getting rid of infected birds when they arise in the farming community. Severe acute respiratory syndrome, I'm sure you've heard of SARS because we just came out of the so-called COVID times, and COVID is a SARS-associated coronavirus, and it produces severe acute respiratory syndrome, and that's what people die of. They die of this inflammatory response in their respiratory system from this virus. Gets in there. There's been SARS viruses before. The COVID disease-causing virus, the SARS-CoV-2, this is actually the name of the virus, has caused a lot of havoc and many millions of deaths over the world in the last 5 years. Acquired immunodeficiency syndrome is the endpoint of infection with human immunodeficiency virus. And this virus just came along in the early 1980s, probably a little before that, probably the 1970s, I believe it is. It's thought to be when it first somehow made the leap from animals in Africa to humans, acquired the ability to infect humans, or did infect humans, and then started producing epidemic or a pandemic. And so we have a lot of problems with HIV for reasons. Again, we'll talk a lot more about that in a later chapter, but it's a very, very terrible disease that is quite dangerous, causes fatality in nearly all cases. Eventually, it's a slow brewing kind of a problem, but it eventually kills. And we've learned a lot about how to treat it, but not how to cure it. And it's a very interesting topic, which we'll talk about later on. Cryptosporidiosis. It's a protozoa that causes a severe diarrheal illness, and it's become a relatively big problem in the United States, transmitted in water, water parks, all kinds of different situations where people can get into it and become infected with it and cause a pretty memorable diarrhea disease. There's some Cryptosporidia oocysts, little eggs that can then germinate and grow into disease. So that wraps up our first chapter of our Tortora Microbiology and Introduction to kick off our microbiology course, and we'll jump into the next chapter immediately hereafter.